Greetings from Microlab Northwest. A few years ago, I made a presentation on pollens, how we identify them, and one of the things that we talked about was using the nutritional supplements, little pollen beads that we can collect from a beehive, and using that as our source of pollens to characterize materials. Now, what we need to do that, before I'd suggested that we use alcohol. We're going to use water today, and we're not going to worry about suspending the individual pellet in alcohol. We're going to suspend it in water. Now you can use just DI water or tap water for that matter, but we're going to need a pipette. Just pick up a little bit of water. We'll put a drop or two down here. So we've got water on our slides. And now I'll use forceps to collect a particular color of pollen. Now we'll pick one that is a dark green, and we'll pick one that is a bright orange on separate slides here. Now we'll mix that up a little bit. I'll just use a nail in this particular case. And the tip of the nail will help us distribute the pollens in the water. And as we distribute the pollen, we're going to end up with a pretty concentrated collection of pollen grains here. So we're going to need to thin down the amount of pollen on our mount that we're going to use to examine the pollens under the microscope. So I've got the green bead here nicely separated. And I'll just use a piece of tissue paper to clean the tip of the nail. And we can move over to the dark orange, the bright orange. Separate some of those pollen grains. Okay, so we've got these nicely separated, good concentration of pollen in the droplet of water on the slide. But now we're going to have to reduce the number of pollen grains so that we have the grains separated so we can examine them individually rather than as a clump. To do that, we'll take one of our plastic pipettes here, and we're going to narrow down the tip. So we light a match and we warm the tip. And we can tell when it's ready because it will go from frosty to clear. And then we can pull that out and wait and watch it become frosted again. When it's frosted, it will retain its position. So now we have a nice pipette with a very thin tip. We'll cut off the tip here that I used to hold it. Now I have a pipette with a very small tip. So I'm going to take another slide. I'm going to pick up a little bit of the pollen here that's suspended in the water, and I'm going to put that on the slide. It's a small drop of what we started with, and now I'm going to add some more water to it. And we'll stir that around a little bit. So we've got our pollen distributed. And now we'll wait and let that dry, and then we'll work on that particular pollen sample. Now when I want to go to the next pollen, I'm going to want to make another pipette so that I don't cross-contaminate the sample. Snip off the end. We have a nice little micropipette. We'll pick up some of the green pollen. Put a little drop of that on this slide. And now we're going to want to add a couple more drops of water. And we'll use our nail that we've cleaned off, spread it around, and we'll let that evaporate, and then we can examine it. Now, rather than sitting here waiting for water to evaporate, I've prepared some slides of these two earlier. So we can take a look at the dark green on the microscope. And you can see that here. Now notice in the dark green, we have fairly large pollen grains. This is fireweed. And here we see a large triporate pollen grain. That's fireweed. There are some interesting things to notice here as we change focus. See that thread-like structure? That's called a vicine thread, and it's characteristic of this particular type of pollen. Those extend out from the pollen grain. Now there are a number of other interesting things that show up here. You'll see this in a live sample of this pollen. You wouldn't see it in an acetylized preparation. And that's all these little particles that you see swarming in the residual water that's still around this pollen grain. These are always associated with fireweed pollen. And it's I think it's a variety of fungal spores, but we'll take a look at that in more detail at a later time. What's interesting here is the fact that this green pellet is all fireweed pollen. And the fireweed, again, a very large pollen grain. It's triporate and has these vicine threads that extend out of it. You might take a look at the dark orange pollens, the ones that come out of the orange pellets. Here under the microscope we can see a couple of them. These, as we mentioned before, are dandelion pollen grains, and as we change focus you can see different characteristics. One of the interesting grains here is the one in the lower right. The lower right grain shows the position of the 3 different pores around the dandelion pollen grain. It also shows us a polar view. We can see what the pole of the pollen grain looks like. The pores are around the equatorial band. Let's go back and take a look at that. In this particular image, we can see the horns that protrude around the pores. There are 2 large horns that protrude around each of the pores. One is just northwest, one is just southwest, and one is almost due west. And as we change our focal position, you can kind of see those 2 horns pointing out. Those are on either side of each of the 3 pores. Now, as we look at the top of the grain, this is the polar section, the polar view. And we can see there is a solid band of the exene that makes up that polar view. Now, as we change focus here, see those dark spots that are suddenly visible? Those are spikes that are pointing out of the grain. They're protruding out of the grain. And because of the sharp angle that's created around those spikes, light can't pass directly through because we're in a higher refractive index mounting media than the refractive index of the pollen grain itself. If we were in lower, those dark spots would look bright. Let's go back and take a look here. Those are nice and dark. We can see where each one of them is located. Now, as we go more toward the equatorial view, we see the 2 horns on either side of the pores. As we go a little deeper, here we can see this girdle of spikes that run between the pores all around the equatorial portion of the grain. Now, that's a particularly good position for us to examine because we can see a lot of the characteristics of this particular pollen grain. If we look at the pollen grain that's in the center, pollen grain in the center is not in such a nice position. We're actually looking at one of the pores, kind of cocked a little bit to one side. We can see the opening of the pore. And as we change focus, we can see that there are spikes in all directions coming out of that pollen grain. Dandelion pollen is very interesting in that respect. So this is a much nicer way of looking at the pollens that come out of our pollen supplements. When we look at the different grains, some of the grains that appear to be the same color might not be the same plant that the bee went to. So taking one of the green ones and then taking a second green one and preparing those, they may or may not be the same plant. That's especially the case with the yellow pollen grains, and the reason for that is lots of flowers have yellow pollen grains. So if you take a different yellow, you'll probably get a different plant. Enjoy your pollen examinations. We'll look at some more pollens in a later video. So long for now.